Hello everyone, in this video I'll be explaining on the topic of gram staining, which is a staining technique done in the lab for diagnosis or differentiation or detection of bacteria in the patient sample. This gram staining is a differential staining used to distinguish and classify bacterial species into large groups that is gram-positive bacteria and gram-negative bacteria. So it is called a differential staining because it differentiates all the bacteria that exist into either a gram-positive bacteria or a gram-negative bacteria. So all the bacteria that exist either falls into either of this category, gram-positive or gram-negative bacteria. And the name comes from the Danish bacteriologist Hans Christian Gram, who was the one who developed this gram staining technique. And so the name of the stain is named after this Danish bacteriologist Hans Christian Gram. And the difference, the principle or the main difference in this gram staining principle depends on the cell wall structure of the gram-positive and gram-negative bacteria. Gram-positive cell wall contains a thick layer of peptidoglycan with numerous teichoic acid cross-linking which resists the decolorization. Gram's iodine acts as a mordant or as a trapping agent, which is enhancing the stain of this primary stain to stain better to the cell wall structure of the bacteria. A mordant is a substance which increases the affinity of the cell wall for a stain by binding to the primary stain, thus forming an insoluble complex which gets trapped in the cell wall. The decolorizing agent, examples such as ethanol or an ethanol and acetone solution, interacts with the lipids of the membrane of both gram-positive and gram-negative bacteria. The outermost of the gram-negative cell, which is lipopolysaccharide layer, is lost from the cell, leaving the peptidoglycan layer exposed. Gram-negative cells have thin layer of peptidoglycan with a slightly different structures than the peptidoglycan of gram-positive cells with ethanol treatment. Gram-negative cell walls become leaky and allow the complexes to be washed from the cell. The highly cross-linked and multi-layer peptidoglycan of the gram-positive cell is dehydrated by the addition of ethanol. The multi-layered nature of the peptidoglycan along with the dehydration from the ethanol treatment traps the complex within the cell. So after decolorization, the gram-positive cell remains purple in color, whereas the gram-negative cell loses the purple color and is only revealed when the counterstain safranin is added. So that is the introduction of gram staining. Here in this gram staining, its main purpose is it differentiates all the bacteria that exist into either a gram-positive or a gram-negative bacteria. And in this staining, the primary stain which will be used is crystal violet and safranin as the secondary stain or counter stain. Why the name secondary stain is given? Because it is a second stain which will be used or poured over the smear or the bacterial sample smear on the slide. And then this staining technique, it is a type of staining technique. A staining technique is a technique which is done in the lab which involves the use of various staining reagents or staining solution which stains the particular microorganisms and then viewed under the microscope where the particular microorganisms can be detected or for the diagnosis of the disease.